today's lesson, we're going to focus on CRISPR and genetic engineering. So the reason why I put genetic engineering, which is 4F, ahead of CRISPR is because CRISPR-Cas9 is a genetic engineering tool. So essentially, if you don't know what genetic engineering is, I don't know why you need to learn CRISPR before that. So I've prioritized genetic engineering first, then we can go into CRISPR. So essentially, this picture here, this is the biggest summary of CRISPR. Looks very complicated, but once you break it down, it's actually step by step, straightforward. Okay, so the things you need to know for CRISPR, one thing is that you need to identify and be able to explain the function of CRISPR and also the features of it. And you also need to be able to understand the ethical concerns behind using CRISPR. So ethical, social, and biological implications. These three classes you need to know and be able to give examples of each, because there are common questions that ask about implications of the technology. Okay, I think I left out genetic engineering out of this, so just be privy of that. So review question, yeah, what is the main function of polymerase chain reaction? And I get okay, if you had to give one sentence to summarize polymerase chain reaction, what would be the function? You could literally say it in 2 words. It will be amplify DNA. Basically, make multiple copies of the same DNA fragment. So we call it amplifying DNA. That's actually the simplest form you can use to define polymerase chain reaction. The mode of action is going to be much more complicated to explain. I will leave that out for now. What are some principles that allow for gel electrophoresis to work? Well, the major one, the one that actually allows them to move from one part of the gel to the other part. What is the property that causes that to happen? Can I get, yeah, why do the fragments move from one side of the gel to the other side of the gel? What causes that? Say it, don't be so scared. Yeah, so rather than just saying the positive charge, you should say the current running through the gel from negative to positive, causing the DNA fragments to move. So now I ask you, right, are DNA fragments positively charged or negatively charged? Can I get Janita? Negatively or positively charged DNA? I'll give you one hint. The fragments are moving towards the positive charge. So what charge does the DNA have? So opposites attract, right? So if it's going towards the positive side, it's the negative charge is attracting it towards the positive charge. All right. Yeah, I think this question is basically just using what I just said. Yeah, glossary task. So let's get into it. Since DNA is the blueprint of life, essentially by changing that blueprint, we can change the thing that's going to come out. So essentially think of a building like this. If I had changed the blueprints of the building right before they were going to build the building, it would have come out differently, right? Same thing with any living organism. If the DNA of that organism has been modified or altered in any way, the final outcome, which is the living thing itself, will also be altered. So essentially, there are 4 ways that this alteration can happen. Is where you insert a gene, you silence a gene, you remove a gene completely. Okay. Remove a gene and then modifying a gene as well. So there are 4 ways that the modification can happen. And essentially, the one that is the most the most important to remember is that adding the gene. That's the one where CRISPR is very useful for. And other forms of modification, like modifying, silencing, and removing, can be done using CRISPR as well. But adding a gene, that's where CRISPR, right? That's one of the things that CRISPR is actually very, very useful for. Okay. Yeah. So using biotechnology, we can modify the genome of an organism. There are four things that can be done. These are done for either commercial or research purposes, right? For example, in commercial purposes, the first genetically modified organism was a tomato where they had silenced a gene that causes it to rot faster. So by silencing that gene, they essentially increased the shelf life of that tomato. It's called a flavor saver tomato, but that's the purpose, right? It was for commercial purposes because we want to be able to keep the tomato fresh for longer, so it's more viable. So there's two forms of genetically modified organisms. One is referred to as cisgenic, the other one is transgenic. This refers to where they're getting that extra gene from. If they are getting it from the same species, meaning that, let's say, an example would be, think of humans that are able to hold their breath underwater for a very long time. If I took one of their genes that allows them to do that, and then I put it into another human, that is a cisgenic organism because I'm taking a gene from humans to put into another human. So it's coming from the same species. So that's why it's cisgenic. If I'm taking something from a different species and then adding it onto another organism, that is referred to as transgenic. An example of that, certain bacteria have the ability to be bioluminescent due to activity of a gene. That gene was taken from the bacteria, inserted into certain organisms, and that caused the organism to be able to produce bioluminescence. In other words, glow in the dark. So this is actually something that is being sold as well. Animals that can glow in the dark, like fish, zebrafish, they were genetically modified with genes from other organisms to glow in the dark, and they would be considered transgenic because it's coming from a different species. That's the two difference. So to know that genetically modified organisms include these both, it's just where the gene is coming from, right? Transgenic, it could come from literally any species other than itself. Cisgenic can only come from the same species. So both are about transferring genes or modifying the genes in any way, but essentially it's talking about where it's coming from, cisgenic and transgenic. Okay, so why does genetic engineering work? So people don't even ask this question a lot, but why is it we can take a gene from some other random organism and then plop it into an organism like us or any other organism and just expect it to work, just expect it to be fine, not cause any troubles? The main reason why this could even be viable is because of the fact that genes are universal, meaning that the codons required to make an amino acid are same for almost all living things. So remember that table that I showed you guys of the codons and the amino acids they make? That is universal for almost all living things. So essentially, if I take the gene, let's say the gene is A, B, G, C, G, A, T, right? And this makes a amino acid. Even if I take this gene and put it into a different organism, it will still make the same amino acids because the codons required to make the amino acids are identical. Okay, that table is identical for almost all living things. So we can expect the amino acids to be produced in the same way, when even regardless of which organism it's going to be in. Following through? I understand what I'm trying to say here. Okay, that is the most important feature of a gene that allows genetic engineering to happen. The rest you still need to remember because of genetics. So there are many techniques that humans have developed to modify genes. Okay, we've been doing this for a very long time, and we've come up with a bunch of different ways. One of the ways that was used before the invention of CRISPR is called a zinc finger nuclease. It's a much more complicated process, which essentially, I don't even want to explain it because you don't need to know it. Okay, it's just one of the techniques, which is a zinc finger nuclease. They are powerful techniques as well, but the invention of CRISPR basically blew all of them out of the water because CRISPR not only is a powerful tool, the key thing that CRISPR does that no other genetic engineering tool is capable of doing is that it is very precise. The CRISPR tool is able to make cuts in the DNA at a very precise location. So think about the endonucleases that we've learned that can make cuts at DNA. So there's restriction enzymes, and restriction enzymes only work at recognition sites, right? They look for specific recognition sites. So example, E, T, C, C, E, C, C, A, right? So let's say, so this could be the entire recognition site, right? Endonuclease will look for this recognition site and then make a cut either as a blunt cut or a sticky cut. That is how a restriction endonuclease works. They are restricted to the fact that it can only make cuts at the restriction sites. But with CRISPR, we can basically program CRISPR to make a cut anywhere in the DNA sequence. As long as we know where we want to make the cut, we can program CRISPR to make a cut exactly at that position. So that's the key feature about CRISPR that no other genetic engineering tool has, is that it is able to make a precise cut at the sequence where we want it to make a cut, which is a very useful tool to edit genomes, which I will talk about in a bit. So depending on the host organism, specific genetic engineering techniques need to be used, right? For example, you can't use the same techniques you would use on a bacteria for humans, right? It might not work the same way. But CRISPR, on the other hand, can actually be used for all living things, right? So that's why it's another reason why it's so useful as a tool, genetic engineering tool, because it is also universally used for almost all living things, and also it can make precise cuts. So some of the genetic engineering techniques that we're going to be looking at is insertion of recombinant plasmids. This is going to be the other topic on recombination, CRISPR-Cas9, which we're going to look at today, zinc finger nuclease, which we won't actually cover, and then insertion of a transgene. All right, so let's start off with CRISPR-Cas9. It's going to be a bit of a complex thing, so stay with me. If at any point you need to catch up, just let me know and I'll go back again. All right. So first thing to note, this technique, this technology, CRISPR-Cas9, is a biological tool that was developed in bacteria. So bacteria actually evolved this tool over many years as a defense mechanism against bacteriophage. So you guys probably haven't heard of the word bacteriophage before. Bacteriophage are basically viruses that attack bacteria. That's it. Okay, they look. I think I have. Yeah, that's the picture. This is the picture of a bacteriophage. You probably have seen it in movies and stuff. They usually show this picture to show bacteria or something like that. So these bacteriophages kill trillions of bacteria every day. And you can imagine this has been going on for many millions and millions of years. So bacteria eventually evolved a system to basically protect themselves from the bacteriophage and what they are trying to do. Essentially, what a bacteriophage tries to do is the same like any virus, what any virus would do. They insert their gene into your genes and make you produce more of them. So essentially, what Corona did as well, the virus invades your cell, takes over the ribosomes, the protein machineries, and command them to make more of the virus. That's essentially what most viruses do. They take over your system and command your system to make more of the virus. What the bacteria did is it somehow created a system that looks through the DNA sequence, finds the sequence that is similar to the virus's sequence, and cuts it out, takes it out of there so that the virus won't get activated at all. Okay, so the original function, like I said, is attacking and destroying viral DNA. So viruses insert their DNA, it inserts itself into the bacterial DNA, waiting for it to be activated. But CRISPR-Cas9 will look through the DNA at the same time searching for any potential viral DNA, and if it sees it, it cuts it out. That's essentially what CRISPR-Cas9 does. So how it works, it all starts with the bacteria saving a segment of DNA from the original bacteriophage. So let's see if I can. Okay, it all starts with the first step. Okay, before I even talk about that, yeah, this will come later on. I'm just going to talk about the prerequisites for CRISPR-Cas9. What do you need to be able to carry out the function of CRISPR-Cas9? The first thing you need is something referred to as clustered regularly interspaced short palindromic repeats, which is the CRISPR, the word. I know it sounds very complicated. That is just referring to a region of the DNA that is going to have repeats and spaces alternating back to back next to each other. So the repeats, they are not that important. The spacer is the important part because the spacer DNA is made up of the copied segment of DNA from the bacteriophage. Remember, like I said, CRISPR-Cas9 looks for the viral DNA in the bacterial DNA, but for it to look for that DNA, it needs basically a reference sequence, right? That reference sequence is going to be at the spacer. These spacers are the copied segment of DNA from bacteriophage that had originally invaded the bacteria. So let me try to put this into an easier way to understand. So think about how your immunity works, right? A virus, a bacteria, or sickness invades you, your body learns to defend itself against it. And then once you get better, your body keeps antibodies to save itself in case the same thing attacks you again. So your body will ramp up the antibodies production and you'll be fine. This is essentially a technique that is called adaptive immunity because your body is adapting to the thing that's invading your body and you're protected from it if it does it again. This CRISPR-Cas9 is a form of adaptive immunity for bacteria. So similar how vaccines work on us, bacteria have this system to basically protect itself from the virus that had originally attacked it. So the virus attacks the bacteriophage attacks the bacteria, and if the bacteria manages to actually survive, it will take a segment of the bacteriophage DNA and put it at the spacer. This is basically protecting itself from the same bacteriophage attacking it in the future. So if that same bacteriophage comes back, attacks the bacteria, the CRISPR-Cas9 will use the spacer and look for that same segment of DNA in the attacking bacteriophage. And if it finds it, it will make a cut there. So upstream of the CRISPR-Cas9 is the gene for the Cas9 protein. So CRISPR-Cas9 is made up of two elements, right? CRISPR, which is the clustered regularly interspaced short palindromic repeats. And then the second element is the Cas9 protein. So the Cas9 protein is made using the Cas9 gene, which is just upstream of the CRISPR sequence. And there's also two other Cas proteins that are involved in the action of CRISPR-Cas9, which you're not going to see here, but you're going to see later on what they do. Essentially, they are bringing the segment of DNA from the attacking bacteriophage to be placed into the CRISPR sequence. Okay, everyone following me so far? So Cas1, Cas2, they put the spacer here. Cas9 uses the spacer to activate the CRISPR-Cas9 complex to find the similar sequence in the attacking bacteriophage. Another thing that's required is the guide RNA, which is basically what happens when the spacer has been transcribed to be used by CRISPR for the system. So we can't just take the spacer and then use it in the system. This needs to be transcribed as a guide RNA, and then that guide RNA will activate the Cas9 protein to be the system that looks for the segment of DNA. The next thing is the protospacer adjacent motif, the PAM sequence. This is basically a reference sequence that the Cas9 protein looks for in the bacteriophage segment, which It's basically simplifying the work of Cas9. I'll explain later more on that when it gets to that. Okay, so now let's talk about how CRISPR-Cas9 works. Okay, try to go a bit slow on this just to make sure you guys don't get lost. So let's imagine a story where a virus is attacking a bacteria. A bacteriophage is attacking a bacteria, right? So the bacteriophage inserts its DNA into the bacteria. The bacteriophage inserts its DNA into the bacteria, and at that very moment, the PAM sequence is recognized. Like I said, right, the PAM sequence is a reference sequence that can be found on the DNA sequence of a bacteriophage. So when it's recognized, the Cas1 and Cas2 proteins are activated, and they will cut out something known as a protospacer. It's called protospacer because essentially it's going to become a spacer, but the word proto means early, right? So the fact that it's only just been cut, that's why it's referred to as a protospacer. It's a new spacer, right? The PAM sequence is left out of this photospacer. This is an important thing to note. It is a recognition site, but it's intentionally left out from the spacer when the cut is being made. So Cas1 and Cas2 will make sure that they cut the sequence, leaving out the PAM sequence when it does that. That is when it is being exposed to the bacteriophage. Okay, once that's happened, the spacer will be integrated into CRISPR and things will be normal again. And then once there's an indication that a bacteriophage is attacking the bacteria again, the CRISPR-Cas9 will be like, I'm going to go through all the library of spacers that I have, making multiple guide RNAs, seeing whether any of them match the bacteriophage that is attacking me right now. So what happens is the CRISPR-Cas9 will be transcribed as guide RNA. Remember, DNA transcribed will become into RNA, right? Transcription, we still remember that, right? Yeah. So CRISPR spacers, which is part of the DNA sequence, are transcribed through transcription to become guide RNA. This guide RNA forms a happy loop to the lagging palindromic repeats. Basically, they also transcribe a bit of the repeat at the back, which causes it to form a hairpin loop. The guide RNA will bind to the Cas9 protein. So remember that there's a Cas9 gene in front of the CRISPR sequence. This will be made into a protein, and that protein will bind together with the guide RNA to become the CRISPR-Cas9 complex. So this is the complex that will do the cutting. This is the system that does the searching and the cutting at the bacterial DNA. Any questions so far? Okay, let me just repeat this again. Exposure. Think of this as you're getting sick for the first time. Like you're having chicken pox for the first time, right? Your body fights the disease and then saves a part of that disease in your body so that if you ever do get sick with it again, your body can fight the disease. Same thing here. The bacteria is exposed to the bacteriophage DNA. It then cuts out the bacteriophage, a segment of the bacteriophage DNA using Cas1 and Cas2 to make a protospacer, leaving out the PAM sequence. The PAM sequence is that recognition sequence for the bacteriophage, and that protospacer will be integrated into the spacer. Once that's done, and it's been integrated, when the bacteria needs to make a CRISPR-Cas9 complex, what happens is that the spacers are transcribed as guide RNA. Specifically, the spacers, remember that the spacers are the ones that are the copies of the bacteriophage DNA. So that's the one we want. So that's the part that's transcribed, including some of the repeat just to create a hairpin loop. And that will bind to the Cas9 protein to create the CRISPR-Cas9 complex. So this is basically you arming the gun, right? It's loaded, it's ready to go. Now it's already has a homing technology, so it just needs to go look for its target now. So now that the stage is set, time for CRISPR-Cas9 to do its work, right? So it all starts by CRISPR-Cas9 complex going to every cell that it can and scanning the entire sequence until it finds a complementary segment to the guide RNA that it has. Because remember that the guide RNA is made using the copy of a bacteriophage DNA. So it's basically going to match, it's going to be complementary to the segment of DNA that the bacteriophage has inserted. So time to make this a bit easier to understand. Remember that bacteriophage inserts its own DNA into bacterial DNA. So at one point of the bacteria DNA, there will be one segment that is viral DNA. So that's the part that Cas9 is looking for. It's like, where is the viral DNA? If it finds it, because it's all complementary, that's where it will make the cut. So when the right PAM sequence is recognized by Cas9, like I said, it's a recognition sequence, but the fact is that recognition sequence was left out of the guide RNA for a reason. It will be recognized by the Cas9 protein. And once it recognizes that, it will see whether everything is complementary here. And if that's the case as well, then the cut is made. So two things are checked. First thing to check is the PAM sequence. Then only it will check whether it's complementary. Because think about this, a DNA sequence is going to be thousands upon thousands of base pairs. The Cas9 protein will take forever if it was checking every base pair to see whether it's complementary. That is where the PAM sequence comes in. It's basically a first check that needs to be approved. Only when it's approved, then it will confirm whether it's complementary. And when both have been confirmed, the cut will be made. If the PAM sequence doesn't even get approved, then the nucleotides aren't necessarily checked whether they're complementary at all. Okay, the PAM sequence is the first verification tool, and then the complementary sequence is the second verification tool. So yeah, it leaves a blunt end. So what does a blunt end mean? Can I get 10? What does a blunt end look like? A blunt end cut. There's only two types of cut we've ever learned, a sticky cut end and a blunt cut end. Okay, so if essentially if a sticky cut leaves a hanging nucleotide, blunt cut will basically just be a straight line. So there will be no overhang. It will literally separate the segment into two different parts. Okay, there's no overhang. It literally creates a space in between the segments. So Cas9 has, so to ensure that both sides of the sequence are cut, because this is DNA we're talking about, it's a double strand, it needs to make sure it cuts on both sides of the strand. Cas9 proteins have two active sites, one on this side and one on that side, and then the cut is made. So once that cut is made, it will look like this, right? It will just be a cut. This does not deactivate anything. It just makes a cut. This cut can actually be repaired. But the thing is, this is inside a bacteria. So the repair mechanisms are going to be for bacteria. So what happens is, when the cut is made, this triggers the DNA repair mechanisms within bacteria to actually try and fix it. Because bacteria doesn't know that this is viral DNA. All it knows is that there's been a cut that's been made in my DNA. I need to fix it. But bacterial repair mechanisms are not going to be perfect. In fact, they are very much prone to error, and they will inadvertently cause mutations to occur at the site of the cut. So these mutations could be anything from insertions to deletions. It really doesn't matter what was the new mutation. Any mutation at all essentially would deactivate the gene, because that's how important, that's how damaging mutations can be. Okay, so these mutations will essentially make the bacteriophage DNA, the segment of bacteriophage DNA, non-functional. And if there is a case where there is a double cut made, but no insertions or deletions or no mutations occurred, there is a potential for the strands to ligase back with no harm. But what happens then? It just repeats the cycle again, because CRISPR-Cas9 is going to just keep looking for it. If it finds it again, it makes the cut again. And the second time, it's more likely to have a mutation, because chance by chance, it will just get a mutation eventually, because CRISPR-Cas9 will keep looking for that complementary bacteriophage DNA. And as long as it can find one, it'll make a cut and then make the repair mechanisms will make it non-functional. Anyone lost so far? Okay, I'm going to go through this very slowly again. So guide RNA, that is I'm trying to think of an analogy for this, but guide RNA is basically the bacteriophage DNA that is saved into the bacteria from a previous attack. Every time another bacteriophage attacks the bacteria, it produces a guide RNA to check whether any of the previous attackers are coming back to attack again. Okay, so it looks for the complementary guide RNA sequence along its own bacteria DNA to see whether there's any complementary sequences. If there are any complementary sequences, what does that mean is that the bacteriophage has already inserted its gene into the bacteria. So what happens is Cas9, CRISPR-Cas9 complex recognizes that, and then it will make two cuts at the double strand, creating a double strand break. When that double strand break has happened, the repair mechanisms within bacteria are activated and they will try to fix it. But since they are not the best, they are prone to making errors, like making mutations in like insertions or deletions. And that essentially will make the bacteriophage DNA that was cut into it to become non-functional. Okay? Anyone confused? That's helpful. Moving on. Okay, so how do we take a system that is basically finding bacterial DNA, a bacteriophage DNA within bacteria, making a cut, and then making it non-functional? How do we take that and make it into a genetic engineering technique? So that's the question, right? So essentially, the things that are changed are, first thing, guide RNA will be artificially made by humans. Okay, we make something called single guide RNA, sgRNA. So that's how we basically modify the Cas9 to look for what we want. Because the guide RNA is basically it's basically like a wanted poster, right? Imagine I give you a wanted poster of someone, you can literally go around looking for that person, right? Same thing, by giving the sgRNA, by making the sgRNA ourselves, we can basically create a target that the Cas9 protein will look for within the sequence until it finds it and then makes cut directly at that point. So basically, we have the power to control exactly where we want to make a cut. That is one of the biggest benefits of the CRISPR-Cas9. Like I said, it can make very, very precise cuts. Yeah, it can make. Another thing is that the Cas9 enzyme with a specific target PAM sequence is obtained. So remember that the PAM sequence is like a recognition site that the Cas9 enzyme looks for when it's searching through the DNA sequence. So since it's searching for the PAM sequence as well, we need to make sure that we modify the Cas9 enzyme with the specific target PAM sequence based on what we're trying to do. If we're trying to edit a part of the gene, we need to make sure that that part, we're accounting for the PAM sequence for that part and making sure that the Cas9 protein comes with that specific target PAM sequence. Another thing is that the specific to the single guide RNA that is used and the target sequence. Oh, that's the CRISPR-Cas9 for gene editing can be used in two ways, right? One is where you make a cut and a new gene is totally inserted in. So create a situation where a new gene can be added into the sequence. So the reason why this can be very useful is that genes cannot just be placed anywhere you want in the sequence to become activated. It needs to be placed in the right spot where it has a promoter in front of it, it has all the necessary regulatory elements around it to become functional. So that's why CRISPR-Cas9 is useful, because we know where we want to make the cut. We just couldn't make the cut before this. But with Cas9, we can make a precise cut at where we want to, and then we just insert the gene there, and it becomes a functional gene because everything else is right already in that location. Another thing it can do is that it can also silence, modify, or remove a gene, right? So essentially, you can see that here. So this is the modified CRISPR-Cas9 complex for human gene editing. So what happens? It makes a double-stranded cut. Now, what can happen here? A part of it can be deleted or a part can be added in. So within the cut, another gene can be added in to either disrupt the DNA or to make a new gene to be produced as well. Or you can, sorry, this is the one where you are, it's a donor DNA, it's being integrated into the DNA. This is different. I'm going to edit this picture, it's not the best. So let me go through the mode of action for how it's used as a genetic engineering tool. So first, the synthetic single guide RNA, which is the synthetic single guide RNA is created in a lab that has a complementary spacer to the target DNA. So essentially, it's looking for the target sequence that we want because we made the synthetic sgRNA. Yes, sorry.
Oh, is that like the
S is single guide RNA. Single guide RNA. Yeah. Essentially, you don't even have to say synthetic because sgRNA is synthetically made. Yeah. So it looks for the complementary spacer to the target. Sorry, it has a complementary spacer to the target DNA. So remember that guide RNA was originally a spacer that has been transcribed and then it folds into a hairpin loop to become a guide RNA. So this synthetic single guide RNA will have the sequence that is complementary to the target DNA. And then once Cas9 enzyme is obtained with the appropriate target PAM sequence, they both will be bound together to form the CRISPR-Cas9 complex. Once that's happened, it will be injected into a cell or a zygote, such as a zygote, right? Because CRISPR-Cas9 will basically go through all the cells, looking through all the sequences until it finds what it's trying to look for. So essentially, by just injecting it into the cell, we are able to make sure that it functions. So the sgRNA mixture, the Cas9 finds the target PAM sequence and checks whether the sgRNA as aligns with the DNA. So same as before, it binds to the DNA sequence, moves along the sequence until the PAM sequence is recognized. The PAM sequence is up to 20 sequences, 3 to 10. Okay, I'll confirm with you how long the PAM sequence can be, but it can vary depending on the specific PAM sequence we're trying to make. You don't really need to know the PAM sequences, but just know that the PAM sequence is the initial recognition that is done by the CRISPR-Cas9. When it recognizes that PAM sequence, then only it verifies it by checking whether the sequence is complementary to the guide RNA. Only when both these verification things have been done, the cut will be made. Okay, so Cas9 cuts the selected sequence of DNA, and the DNA has a blunt end. And when repairing the DNA, the cell may introduce new nucleotides into the DNA at the site. What we do is we may inject particular nucleotides sequences into the cell so that it will be accepted into the gap. So we also inject it with DNA ligase. And what is the function of DNA ligase? Can I get Yasmin? What does DNA ligase do? Why is it important when we're doing this? The cut has been made. When we are inserting the gene of interest, we need to include DNA ligase with it. Why? What does ligase do? DNA ligase. Okay. So just because we've inserted the gene of interest doesn't mean they will just stick together with the sequence, right? We need an enzyme that can actually stick them together, the target sequence and the other, the broken donor DNA. So DNA ligase will make sure that they get combined together. Yes. For Cas9, they always make blunt cuts. They do not make sticky cuts. Yeah, sticky cuts are very much a restriction endonuclease thing. Yeah, I have never heard of any other thing making a sticky cut other than a restriction enzyme. So remember that restriction enzyme, restriction endonuclease, they're the same thing. You can use them alternatively. So Cas9's CRISPR-Cas9 complex, they make a blunt cut. The gene of interest is floating around with DNA ligase floating around. Eventually, it will find itself in the right position for ligase to stick together the gene of interest in between the gap, creating making sure that the gene of interest has been integrated into the DNA. So now there's a gene ready to be produced that is correctly placed where it needs to be in the DNA sequence. Okay? I know this is all very heavy, so let's watch a video to bring it down a notch.
From the smallest single-celled organism to the largest creatures on Earth, every living thing is defined by its genes. The DNA contained in our genes acts like an instruction manual for our cells. 4 building blocks called bases are strung together in precise sequences, which tell the cell how to behave and form the basis for our every trait. But with recent advancements in gene editing tools, scientists can change an organism's fundamental features in record time. They can engineer drought-resistant crops and create apples that don't brown. They might even prevent the spread of infectious outbreaks and develop cures for genetic diseases. CRISPR is the fastest, easiest, and cheapest of the gene editing tools responsible for this new wave of science. But where did this medical marvel come from? How does it work? And what can it do? Surprisingly, CRISPR is actually a natural process that's long functioned as a bacterial immune system. Originally found defending single-cell bacteria and archaea against invading viruses, naturally occurring CRISPR uses 2 main components. The first are short snippets of repetitive DNA sequences called clustered, regularly interspaced short palindromic repeats, or simply CRISPRs. The second are Cas, or CRISPR-associated proteins, which chop up DNA like molecular scissors. When a virus invades a bacterium, Cas proteins cut out a segment of the viral DNA to stitch into the bacterium's CRISPR region, capturing a chemical snapshot of the infection. Those viral codes are then copied into short pieces of RNA. This molecule plays many roles in our cells, but in the case of CRISPR, RNA binds to a special protein called Cas9. The resulting complexes act like scouts, latching onto free-floating genetic material and searching for a match to the virus. If the virus invades again, the scout complex recognizes it immediately, and Cas9 swiftly destroys the viral DNA. Lots of bacteria have this type of defense mechanism, but in 2012, scientists figured out how to hijack CRISPR to target not just viral DNA, but any DNA in almost any organism. With the right tools, this viral immune system becomes a precise gene editing tool, which can alter DNA and change specific genes almost as easily as fixing a typo. Here's how it works in the lab. Scientists design a guide RNA to match the gene they want to edit and attach it to Cas9. Like the viral RNA in the CRISPR immune system, the guide RNA directs Cas9 to the target gene, and the protein's molecular scissors snip the DNA. This is the key to CRISPR's power. Just by injecting Cas9 bound to a short piece of custom guide RNA, scientists can edit practically any gene in the genome. Once the DNA is cut, the cell will try to repair it. Typically, proteins called nucleases trim the broken ends and join them back together. But this type of repair process, called non-homologous end joining, is prone to mistakes and can lead to extra or missing bases. The resulting gene is often unusable and turned off. However, if scientists add a separate sequence of template DNA to their CRISPR cocktail, cellular proteins can perform a different DNA repair process called homology-directed repair. This template DNA is used as a blueprint to guide the rebuilding process, repairing a defective gene or even inserting a completely new one. The ability to fix DNA errors means that CRISPR could potentially create new treatments for diseases linked to specific genetic errors like cystic fibrosis or sickle cell anemia. And since it's not limited to humans, the applications are almost endless. CRISPR could create plants that yield larger fruit, mosquitoes that can't transmit malaria, or even reprogram drug-resistant cancer cells. It's also a powerful tool for studying the genome, allowing scientists to watch what happens when genes are turned off or changed within an organism. CRISPR isn't perfect yet. It doesn't always make just the intended changes. And since it's difficult to predict the long-term implications of a CRISPR edit, this technology raises big ethical questions. It's up to us to decide the best course forward as CRISPR leaves single-celled organisms behind and heads into labs, farms, hospitals, and organisms around the world. Wrap your head around the implications of CRISPR with this playlist on genetics, or check out some of the other mind-blowing science being developed around the world and in the universe right now.
Right. All right. So now that we know how CRISPR works, how we use it for genetic engineering, let's think about what are the applications, right? How can it actually serve a function for us? So one thing, for research purposes, we can actually attach specific identification markers to DNA sequences. When I say identification markers, basically fluorescent proteins. So we know for a fact that if we insert this gene at the right place, how to say this? Let's say this is one region of a gene, right? And we want to see whether gene G, not going to say gene G. Okay, gene A, we want to see whether gene A is being activated or not. So how can we do this in the best way is placing another gene nearby it, right? Let's say I place a fluorescent protein gene right before the A. What happens is when there is a transcription of A trying to happen, it will also transcribe B. So what happens is that this protein will be expressed along with A as well. So it acts as a marker to show that A has been activated. So using CRISPR, we can insert these markers wherever we want to, so we can find out whether specific genes are being activated or not. That's what a point of marker is. Another thing, we can disrupt expression of genes to see what it might do, right? We are still very much studying a lot of the genes that we have discovered. We still are very much discovering what their properties are, what knocking them down can do. So using CRISPR-Cas9, we can actually study in much more detail the effects of deactivating certain genes and so on. Another thing you can do, dealing with diseases. So essentially, you know how CRISPR-Cas9 can make a very precise repair? There are over 200 diseases, that's just 200 diseases that are caused by literally one nucleotide being wrong in the sequence, right? Just because this one nucleotide is wrong, it causes a disease. Some of them, Huntington's disease, yeah, there's a lot that are caused literally because there's one nucleotide wrong in the gene that's supposed to be made. By using CRISPR-Cas9, you can say goodbye to all of those. All 200 of those diseases can be fixed literally using the CRISPR-Cas9 because it's that simple to fix them. Because all you have to do is replace one nucleotide and you can fix the disease. So that is one of the amazing technologies that CRISPR can do. Amazing things that CRISPR can do. Another thing is introducing, so basically anywhere you modify the gene to benefit us, right? So another example, introducing pest and herbicide resistant genes into crops, right? So if we know the genes for pest resistance and herbicide resistance, we can insert those into crops to make sure they are more resistant so that we benefit in the long run. Another thing, altering plant genomes to make them grow faster, produce better food, more nutritious food. Honestly, with genetic engineering, the applications are limitless because we can modify life to whatever extent we want. We can think about humans, right? Think about all the things we can modify within our cells using CRISPR, right? Increase intelligence, increase muscle density, increase height, increase anything you want, decrease your, I don't know, like whatever you want to do in terms of biologically modifying an organism is now somewhat possible because of the technology of CRISPR, because of how capable it is to edit genomes. We are actually able to think about where this technology can actually lead us. Some of the limitations for CRISPR-Cas9 that are still prevalent today. For one thing, this scientists must introduce nucleotide sequences carefully to make sure that it will be absorbed by the DNA, right? DNA sequence, so that it has been modified. But this process isn't always perfect because this is more about random chance. The DNA ligase and the target gene need to be at the right place at the right time for it to be integrated into the target, into the donor DNA, sorry, into the DNA sequence that we're trying to edit. So it can be difficult to achieve with precision and is not always successful. Another thing is that to successfully alter an entire genome's DNA, right? So every cell in your body, if you want to make sure it's been altered, it needs to be done at a very early stage. Basically, when you are basically a clump of cells, specifically as an embryo. So when you're an embryo, if your genes have been edited, you will basically grow up with those edited genes. But if I currently, right now, try to edit a region of your body with edited genes, only that region will have it, and over time, all those modifications will also be removed because your body knows what its original DNA is, and it will over time remove the foreign DNA. But if you alter the DNA at that very early stage, where it's still only 10 or 20 cells that are making up the entire organism, then you have a potential to create an organism that is completely genetically modified, right? So one of the problems with that is that some groups are concerned scientific research on embryos does not respect the sanctity of human life. So essentially, if we can modify anything we want with a child, doesn't that kind of make it, the sanctity of life loses its value if life itself can be modified to be whatever you want? So in the same way that having the cheat codes for a video game makes the video game pointless, if you can edit anything you want about a human life, then the sanctity of the human life itself loses its value. In addition, it is also very much currently illegal to implant genetically modified embryos into human females. There has never been a genetically modified baby that has been born, and I don't think that's going to happen for a while because there's still a lot of social, what's the word, backlash regarding doing something like this. So is anyone confused about why I said this? To alter an entire organism's DNA, it needs to happen at the very earliest stage because that's when you're a few cells and you still haven't basically what's the word? Developed into a fully grown organism. Remember that those 10 cells are the ones that will divide continuously to become the trillions of cells that you are today. So if I've edited those 10 cells to be different, then every cell that comes after that will be different in terms of having the target gene. So some of the ethical concerns. Well, safety, obviously, the possibility of off-target cleavages, edits in the wrong place, and mosaics, some cells containing edited genomes, others not, means that many scientists hesitate to use CRISPR outside of research. So there's two things that can happen, right? One is that the edits can happen at the wrong place because DNA sequence is so long that there will be, by random chance, exact same sequences happening at different places. Sometimes they can be Sometimes they can be due to genes that have been copied over to different parts of the sequence. Sometimes it can be just by random chance that that specific sequence that the sgRNA has is found in another place. So it can happen where it's off-target cleavages, but since it also makes sure to cut at the right place, that's still not too bad. Another thing is informed consent. How do you get the consent of an embryo whether it wants to be edited or not, right? You can't. So that's a problem. Next is inequality, right? This kind of a technology will obviously be accessible to a developed nation first rather than a developing or a undeveloped nation. So think about the technologies that we would have access to. Modifying crops, giving kids higher life expectancies, increasing intelligence, all these kind of things, it would present in an inequality because only the developed nations are able to do that. Next is discrimination. Obviously, discrimination will happen where, oh, you're a modified organism. I'm not a modified organism. I'm better than you. Obviously, that would happen. Okay, so what are some ethical approaches involving CRISPR-Cas9 gene editing? So this brings a lot of discussions in regards to ethical approaches. So remember those ethical approaches that we looked at the start of your course: consequences-based, duty-based, duty rules-based, or virtues-based. CRISPR-Cas9 can be argued in each of these, and you need to be able to relate them to each of these in different contexts because they will probably be asking these kind of questions relating to CRISPR. Because there's a lot of ethical concerns relating to CRISPR, so chances are there will be questions relating it to that as well. So remember that consequences-based is where consideration is given to whatever ends justify the means. So meaning if the end benefit is greater than the cost. So if I can save 1 billion lives tomorrow, I don't mind killing 1 million lives today. That is what consequences based is trying to say. I don't care what I have to do today if it can bring more benefit tomorrow. Okay, duty or rule based. This is where consideration is given to social duty or following rules. So essentially, I have a social duty to do this for the benefit of humanity, right? That would be under duty or rule based. Another one is I'm following rules. So that would essentially not be considered here, but you could think of it. Virtues-based consideration is given to the virtue or moral character of a person carrying out the action. So if my intentions when I was researching and developing these technologies are good, that would fall under this. I'm trying to create a technology that can help humanity. That is my virtue. That is why I'm doing this. So that could be a consideration. Where am I coming from? Why am I trying to do this? Oh, okay. This is going into genetic engineering. Essentially, this is another technology for genetic engineering. This is the insertion of DNA, direct insertion of DNA through bacterial plasmids into plant cells. So this is another way genetic engineering can be done. And this is specifically in agricultural plants. This is a common one that's used until today as well, where genes are literally inserted into plasmids and grown in a petri dish until it cultivates the gene. And then once that has been done, we grow them up into plants. So let's go start first. Particular gene of interest is isolated from another organism. So whatever gene we want, we first isolate it and take it from the organism we want. Usually it's a beneficial gene that gives some kind of useful trait like herbicide resistance or bacteria resistance or less reliance on fertilizers, improved drought tolerance. There's so many things that you could try to look for that genes have. Once that's been done, we need to take that gene and insert it and deliver it into the organism. So what happens is isolated genes are inserted into the cell through direct insertion, where using bacterial plasmids. So you guys haven't learned bacterial plasmids, have you? Okay, essentially bacterial plasmids are bacterial DNA. So if this is one bacteria. There's chromosomal DNA here, but there's also one more form of DNA, which is a circular DNA, and we call that plasmid DNA. Okay, this we'll be looking at that in a recombination. But this plasmid DNA is a functional DNA copy that exists separate from the chromosomal DNA. Okay, so it functions similarly as the chromosomal DNA. It's just shorter and can be actually used for genetic engineering as well. So what happens is the genes are inserted as bacterial plasmids into the DNA, into the bacteria. And because bacteria already has plasmid DNA inside it, it won't think of it as being a foreign DNA. It will just accept it as being another plasmid within itself. Because bacteria have multiple plasmids throughout it. So by inserting one plasmid into the bacteria, we can possibly make the bacteria accept it. So that is what bacterial plasmids are, right? So by inserting this bacterial plasmid directly into the plant, they can be grown repeatedly using plant tissues, and then eventually they will be able to be grown into a fully plant. Sorry, suddenly lost. Okay, so essentially, particular gene of interest is isolated. That gene of interest is placed into a bacterial plasmid. That bacterial plasmid is inserted into a plant cell, and we are going to culture those plant cells to see which of those plant cells has actually accepted that bacterial plasmid and is actually expressing it. Eventually, we find a cell or find clumps of cells that are actually producing the gene of interest. And then once we have those, we cultivate that plant cells until we get a fully grown plant that is basically going to be genetically modified. Who's confused about this? Okay, step again. Okay, let's just use the pictures this time. Okay, don't think about anything else. First of all, gene of interest is isolated from any organism we want. Whatever gene this is, it has a benefit, so we isolate it first. We take this and make a bacterial plasmid using that. Okay, bacterial plasmids are basically circular rings of DNA. That's all you need to know. They're just circular rings of DNA. And we make a circular piece of DNA using the gene of interest, and then we insert it into the cells of a plant cell. Sorry, we insert it into a plant cell. That plant cell can either uptake the gene or it can just not take it at all. So to know whether the plant cells have taken up the gene, we need to basically culture the plant cells over and over until we see one cell that is actually producing the protein of the gene of interest. So if it does produce that gene, we know that the plant has taken up the bacterial plasmid. Once we know that, we basically want that cell to be grown into a fully grown plant. Then we know we have a genetically modified crop. There's no way we're finishing this. There's 9 more slides to go. Okay, genetically modified organisms. So as soon as you modify the genome of a living thing, it's become a genetically modified organism, as we looked at just now. So for any genetically modified food or organism to be basically produced as food for human consumption, it needs to go through rigorous testing. So you guys probably don't even realize, but there's so much of the food that you're eating now has been already genetically modified. It's become a very common thing to do to genetically modify your food to give it certain traits. For example, you can see here, right? In soybeans, some of the traits that have been introduced, herbicide resistance and increased oleic acid. In tomatoes, delayed ripening, like I said, and salt tolerance. In potatoes, virus resistance, reduced asparagine, cotton, so on. So you can see that there are a lot of traits that can be added on to plants or animals or anything using genetic engineering. But specifically for these things, these are crops that we want to grow. How can we make those crops better for us? By doing these things, right? By making sure they're herbicide resistant, we don't need to spray our food with herbicide. That's going to be beneficial for us. By making sure it has more nutrients, we benefit from it. by making sure it has salt tolerance. That's new. I didn't know what that is. So everything we do, every modification we do is going to be because it's leading to a benefit for society as it is. That's why we would even do the thing in the first place. No one makes a genetically modified organism for the heck of it. There has to be a benefit. Unless it's just specifically for commercial purposes, like making glow-in-the-dark zebrafish or research purposes. Yeah, this is because usage of GMOs in everyday life can have biological, social, and ethical implications. So there are different types of GMOs that have traits depending on the needs of the people, like I said, right? So there are two types that we need to discuss specifically. These are the ones that probably will be included in your test. Okay. There are crops that have been modified such that they have an increased crop productivity. There are crops that have been modified such that they are more disease resistant. Okay. So by increasing the crop productivity, what's happened is that the crops will make more food per unit of farming land. Essentially, another thing we can do to it is also increase the nutritional value. So if a grain of rice had 5 grams of carbs before, just imagining, by modifying it, we might be able to make it have 7 or 10 grams, right? Essentially, that's what it is to modify the nutritional values. For crops that are more disease resistant, we can make them less impacted by harmful plant pathogens. So we can also make them resistant to environmental factors like droughts and herbivorous pests and herbicide and so on. So whatever modification we do for crops specifically, it's only ever going to be for the benefit of society, right? Crop yield or disease resistance or making sure it is producing a certain nutrient. So one of the examples is golden rice. So golden rice is a genetically modified organism, genetically modified crop, which is technically considered as a genetically modified organism that is developed in response to vitamin A deficiency in developing nations. So what this commercial company did is that they made this golden rice to have 2 genes inserted into its genome where I'm not going to go through these genes, specifically the Psi gene and the CRTI gene. These are coming from different organisms. So what type of what kind of genetically modified organism would this be? Transgenic, exactly, because it's coming from different organisms. So these genes essentially store beta carotene, a precursor of vitamin A, within the rice grains itself, then in the leaves as normal rice would. So essentially, normal rice has this beta carotene in its leaves. By modifying the genes, we've caused it to store it in the rice instead. So that essentially will increase the vitamin A content within the rice. So this precursor molecule is what gives the rice the golden yellow color. And then, so yeah, essentially, if you can look here at this picture, you can see that if I asked you where rice would be, the answer would be 0, because rice doesn't have any vitamin A inside it. But by modifying the genes to store beta carotene inside the rice itself, we've increased the vitamin A content to 3.57 micrograms per gram. Another example is, why did I not make this clear? So like I said, right, these two are the examples that you guys need to know. Crops that have increased crop productivity and crops that are more disease resistant. So the first one, this is the one talking about changing its nutritional content. And this is the one talking about disease resistance, right? So BT crops, these are very common all throughout the world now, where it's developed in response to pest destroying crops. So they inserted the crystal toxin gene into the genome of the targeted crops. And what it does is that this gene